guys, welcome to Med Sense, where everything makes sense. Today we are going to be treating another very interesting topic today called the Krebs Cycle. Why do we call it the Krebs Cycle? This is because it was discovered by a scientist called Hans Adolf Krebs in the year 1937. So we call it the Krebs Cycle. Equally, we can call it the tricarboxylic acid cycle. Equally, we can call it the citric acid cycle. So we're going to look at the meaning of all of these terms later on as we dive in. So let's ride on. Fine. Now, today, we are going to look at the Krebs cycle. And we know that we left the other cycle, the other pathway, which was the glycolytic pathway. If you haven't watched the glycolytic pathway, please go and check our glycolytic pathway and you will understand what it means. Now, I guess that if you must have followed the other video, you notice that we use glucose, for example. And now from this glucose molecule, we are able to synthesize a particular molecule called pyruvate. Let me use this. We had pyruvate. Cool. Now, what is the structure of this pyruvate we saw last time? You'll notice that our pyruvate molecule was something of this nature. Here we have this, and here we have something like this. Cool. Now this is how our pyruvate molecule looks like. This molecule now, it is about to enter the Krebs cycle. Remember that glycolysis is taking place where? It is actually taking place at the level of the cytoplasm. That is where it is taking place. Next thing, where is the Krebs cycle, which is what we are looking at now? Where is it taking place? It is taking place in the mitochondrion of the cell. So we are at the level of the cytoplasm of the mitochondria, which is called the organic matrix. That is where we are going to be moving into. But now this our pyruvate, which we had formed, cannot actually enter the membrane cannot penetrate through the membrane of the mitochondria. So it needs to be converted to a nice molecule that can actually go through the membrane of the mitochondria with ease. What is that molecule? That molecule is called acetyl-CoA. That is what we are going to be forming from this our pyruvate. And when we are forming acetyl-CoA, we need something of this nature. We are going to be using pyruvate. We are going to add what we call coenzyme A, CoA. So we are going to add CoA with a sulfur hydroxyl group. And now, from this compound, we are going to remove this carbon dioxide. There will be decarboxylation in this case. So I'm going to remove carbon dioxide. Equally, what is going to happen? I'm going to use a very nice enzyme here called pyruvate. This enzyme will be called pyruvate dehydrogenase. Why? Because I'm going to be removing hydrogen from the compound, where I will use NAD plus to form NADH. So when you get off dehydrogenase, start thinking of what? Start thinking of the enzyme dehydrogenase. And in this case, since we are dehydrogenating pyruvate, should be pyruvate dehydrogenase. Cool. Now, what is going to be the new substance, or let me say, the new compound formed in this case? We are going to form another compound in this case, which is going to be called acetyl-CoA. This is our acetyl-CoA formed. What is the chemical structure of acetyl-CoA, so that everything makes a lot of sense? Our structure for acetyl-CoA is going to go as such. We are going to have this. Remember, we have removed this group here. We are left with something of this format. So if we are left with something of this format, our hydroxyl group is going to attach itself to this carbon atom here. So I maintain what is there. I put this, and then I have this. We have our CoA. Cool. So our hydrogen that has been removed from this compound is the one with which this man here enters with. So this is going to be our new compound form. This is how acetyl-CoA looks like. Now, acetyl-CoA is very, very interesting. The guy now can move into the mitochondria. And when it moves into the mitochondria, it is supposed now to combine with another substrate in there so as to keep running the cycle. But which one is that? That is the question right now. Do you know we have a particular substrate here? We call this substrate here oxalate. We call it oxalate. So we are going to have oxalate like this. This is how our oxalate will look like. Let me put it here. We have oxalate. Now, oxalate, how does it look like? That is the question. How does it look like? Oxalate here. Okay. Now the next thing is the chemical structure of oxalate. You know, oxalate is a 4-carbon compound, and it is a dicarboxylic acid compound equally. So we are going to always have this, and then you have COO minus bonding to COO minus. Then here I have this compound of mine, CH2. This is how oxalate looks like. Now, this guy here is going to combine with oxalate, because automatically once it combines with oxalate, it is going to join this cycle at this point. Our acetyl-CoA combines with oxalate. Once it combines with oxalate, what happens? There's something very interesting here. Water is going to be added into this. We have our water. The next thing, remember that if this guy is combining with oxalate, how many carbon compounds do we have here? 1, 2, 3, 4, 5, 6. We are going to form a 6-carbon compound, and that 6-carbon compound is called citrate. And by doing so, what is going to be happening again? I'm going to be removing my CoA with the sulfur hydroxy group. That's another thing that is going to happen here. So in that case, this man has combined with oxalate. We are going to form our 6-carbon compound. This 6-carbon compound here is called what? That is the question. It is called what? The 6-carbon compound here, if I should arrange this, we used this color rather. Let me arrange it to look in this manner. Very good. Okay, fine. We said we are adding water. We said we are removing what? Our CoA with the sulfur hydroxy group. Now, which enzyme are we using in this process? We are going to use a very nice enzyme called, remember we are forming a 6-carbon compound called citrate. So we are going to call this enzyme here citrate synthetase. Cool. Now, the next thing, we are going to have this our compound here. This compound here will be called citrate. Our compound here is called citrate. The next thing, how do we have the chemical structure of this compound called citrate? This compound called citrate, remember I've combined these two compounds, so automatically I'm adding water. If I'm adding water, I'm hydrolyzing this bond. So automatically everything should come back as such. We are going to have this, and then here, remember we are going to be lysing this bond to form an OH group here. And then here, remember, I will just carry this group here and attach it here. So I need to make it to look some kind of like differentiated from this group and this group. So in that case, I'm going to put the other group in red so that everything makes a lot of sense to us. Remember, we still have this carboxy group here, like this. And then from here now, we are supposed to have this carbon atom here. This is our carbon atom that is linking to this group. And then from the upper one, as we are removing this group from this compound, there is carboxylation of this carbon atom. So much such that I have something of this nature. And then automatically I bring back my 2 hydrogen atoms. Okay, cool. Now, We are moving to the next step. So this is called citrate. You know I'm going to carry out an isomerism reaction in this case. Which type of isomerism is here? Position isomerism, where I'll be changing the position of this OH group to this carbon atom here. That is what I'm actually going to do. And when I'm doing so, I'm forming an isomer of citrate. This isomer of citrate will be called isocitrate. Isocitrate. Now, this process, I will need to use a particular enzyme. This enzyme is called the aconitase enzyme. That's the function of this enzyme. So how will my chemical structure look like? My chemical structure, remember we are moving from here, we are transferring this group back here. So I should have something of this manner. I'll have something like this. OH, I'll have my COOH, my COO group, and then here I have my hydrogen, and then here I bring back my COO minus. So that is what is taking place here. And then back here I replace back my normal group here. This group, remember, where is it coming from? It is coming from our normal acetyl-CoA, which we had over there. So this is isocitrate. So what is the change here? Simply the OH group is the one that has changed position. We are moving to the next step. Let's ride on. Everything is cool up to this moment. I guess so. Okay, we are riding on. Now I need to convert from isocitrate to another compound in which the number of carbon atoms is supposed to change. Remember, here we have a 6 carbon atom compound. The number of carbon atoms, they are supposed to change from 6 to 5. So what is supposed to happen? Don't you think I'm supposed to remove another carbon dioxide? Cool. That is what is actually going to happen in this case. So I should be using this mechanism here, where I will form a new molecule called alpha ketoglutarate. Alpha ketoglutarate. How does it look like? What am I doing? Because actually at this level, what I'm simply doing here is I'm carrying out decarboxylation. We have decarboxylation. Another thing, I'm carrying out dehydrogenation. And if I'm carrying out dehydrogenation, I should use a particular enzyme here. What am I dehydrogenating? Isocitrate. So it should be isocitrate dehydrogenase. That is my enzyme here. I'll call it isocitrate dehydrogenase. Cool. Now, what am I doing in that case? If I'm carrying out dehydrogenation, therefore, I will need a hydrogen carrier, which in this case, it is NAD+. So we are going to notice that NAD+ is going to be reduced by hydrogen to NADH. Very cool. That's what is going to be happening in this case. If that is what is happening here, then what will be our new compound? How will it look like? Our new compound here is going to be something of this sort. Remember, I remove hydrogen from here. Sorry, this compound is incomplete. We are supposed to have hydrogen at this point. This is how it's supposed to look like, right? Now, here I remove hydrogen from here. I'll be left with this. And then my new compound, this hydrogen now flips up so much such that I have this. I have removed this carbon dioxide from here. I've removed carbon dioxide from here. So what I'm left with now, it is simply this compound. That is what I'm left with. I replace back the carboxyl group, which is supposed to be here. The next thing, I'm supposed to move and replace back this my group, which came from my acetyl-CoA. Don't forget, remember. That is what I'm supposed to have here, and then 2 hydrogen. There are 2 of them, actually. Now, what next? What is the next thing we are supposed to be doing here? We are supposed to carry out the next step. If this is actually decarboxylation and dehydrogenation taking place here using isocitrate dehydrogenase, I've formed this new compound called alpha ketoglutarate with 5 carbon atoms. Now, I'm supposed to move to the next thing. That is cool, isn't it? I'm supposed to form a new compound here, which is called succinyl CoA. Succinyl CoA. Oh, what the heck is this name? Where is it coming from? Therefore, that means I am adding a CoA with a sulfur hydroxy group in this case. So that is what is happening in this case. So how will it look like? It is going to look like this: CoA with a sulfur hydroxyl group. That's how it is going to look like. And then upon that process, I'm going to be carrying out dehydrogenation and decarboxylation again. Oh, so I'm going to be carrying out decarboxylation, removing out the carbon dioxide, and then I'll be carrying out dehydrogenation. Remember, when we talk of dehydrogenation, think of NAD+, which we are going to reduce it to the form NADH, isn't it? Now I'm going to use a specific enzyme here. What is this enzyme? Since I am removing the hydrogen from alpha ketoglutarate to form succinyl CoA, I should use the enzyme alpha ketoglutarate dehydrogenase. Cool. Now the next step, we are going to be looking at the structure of succinyl CoA. I've removed this carbon dioxide in this case. This is the carbon dioxide I'm removing in this case. So how should my new structure look like? It should be this. I've removed this group so much such that it can be substituted by this, our CoA here. So this, our CoA with the sulfur hydroxy group. The sulfur group will attach here. And then what is going to happen here equally? I'm going to have now my CoA group here. Cool. Now, what next? I'm going to have this carbon atom. I put it, right? The next thing, what am I going to have here? I replace back this my H2, and then I come here where I'm going to have my two hydrogens bonding to this carbon atom with my COO minus group. Now, we can see that that compound is quite cool. From here, I'm supposed to carry out again something. We use our blue ink to go to the next step. So I'm leaving from succinyl CoA, which has this chemical structure. From this succinyl CoA, I'm going to form a new compound, and this new compound is called succinate. I'm going to call it succinate. Called succinate. So I'm going to call it succinate. How does succinate look like? Succinate, now remember, whenever I add this my CoA with a sulfur hydroxy group, in the next step, what am I going to be doing to it? I'm going to be removing it and then carboxylating the carbon atom to which it was found. That is the only thing we are going to do here. Isn't that cool? That is quite cool. So I'm going to have something of the form. Remember, I carboxylate this carbon atom. And then what next? I have my carbon atom here with the two hydrogen. Then next, what do? I have this other compound here. Now, cool. So this is the compound we have here. This is quite succinate. But how did I get to succinate? That is the question from here. I'm going to be having a very interesting reaction taking part here. That means I will be forming, let me say, I'll be using GZP to form GTP. Now, when I do this, that means I have phosphorylated this GTP. And now there is a very interesting reaction that is going to take place here. Another person will come in here, such as ADP, and say, hey, I need this phosphate group, give it to me. And then it becomes ATP. That is cool. So ever since we began here, this is where we are forming ATP since we started this cycle. Now, that is what is going to happen here. And what next is going to be taking place here? We're equally going to have the removal of this sulfur hydroxy group. So I'll be removing this sulfur hydroxy group, the CoA sulfur hydroxy group. I'll be removing it. After having removed it, I carboxylate this carbon atom. That is why you have this. Sorry, it is not going to look like this. Going to look like this. Cool. This should be the right way, isn't it? So that was a mistake. When we are carboxylating this carbon atom, it should look like this. Now, we are moving to the next step. We are leaving succinate. When we are leaving succinate, we are moving to another interesting compound. So this is how succinate looks like. If we are leaving succinate to another interesting compound, this compound is going to be called fumarate. And when I'm forming this compound called fumarate, a very interesting reaction is still supposed to take place here. I will carry out dehydrogenation here. But the dehydrogenation is special, not as the one we have been seeing before. We are going to be using FAD+. And this FAD+ we are going to be using during dehydrogenation. I'm going to have FAD+ here. To form what? To form FADH2. Oh, haven't you noticed something? Instead of removing just one hydrogen, as in the case of NAD, here I'm removing two. So where are the two hydrogens coming from in this compound? That is the next question. So I'm forming fumarate. That's what we said, right? I'm forming fumarate. Cool. Now, I'm carrying out dehydrogenation. That means I'll remove one hydrogen from here and one from here. That is why I'm having two of the hydrogens. So how will the chemical structure now look like? It is going to look as such. We are going to have this. Remember, we have removed one hydrogen from here. We have this. We have removed also another hydrogen to be left with one. But then, remember, this structure is not to be written in black. So let's use our normal color. So I'll have this. So remember my hydrogen. Fine. Now, as we can see, in between these two carbon atoms, please, there's something which we are missing out here. Remember that whenever I've done this dehydrogenation, I have to have a double bond here because a carbon atom must have 4 complete bonds. Fine. So here I have formed my fumarate. Through what process? Through the process of dehydrogenation. So from succinate, I have formed this guy. Are we there? So that is actually what is taking place here. What else? What next? That is the next question. So we move to the next thing. From fumarate, I'm going to form another chemical compound, which is going to be called malate. I'll form malate. Now, from malate, I'm supposed to have this. From malate to this, I'll need to have this. And now, something is taking place here, very important. That means from here to here, I am adding water. That is what is happening. I am adding water. And if I am adding water, that means I'll have a new chemical structure. I'll have an OH group coming in here. How? Because remember, how does water behave? Remember that water, whenever it is splitted, like this, whenever splitted, it forms the hydrogen ion and the hydroxide ion. Now, when you have this, our new compound, how will the change look like? We are going to have something of this sort. Remember, this is our group, right? Now here, this hydrogen will come to this compound so much such that we have this. Next, this double bond here is supposed to be broken down by the water coming in. It breaks it to fit in, right? So once it breaks it to fit in, now we are going to have just this single bond. And then remember, we are having our hydrogen here, and then we have our OH group from here, the splitting of that our water. So this is our water. We are fit in hydrogen here to have this. We are fit in the OH group here to have this. Now, then what next? We have this equally. So that is what is taking place here. Now, if this is how my malate looks like, I need to leave from this compound back to this compound. Isn't that cool? I'm supposed to leave from this compound back to this compound. If I'm to do so, what is supposed to take place? What is going to happen in this new compound? That means I'll be carrying out another decarboxylation, and dehydrogenation will also take place here. Don't forget. So, if we have 1, 2, 3, 4, here we have 1, 2, 3, 4. Sorry, there's no decarboxylation, we just have dehydrogenation. That is a mistake. Okay, we are going to have from here to here, we are going to have what? We are going to have dehydrogenation. And if I'm carrying out dehydrogenation, what am I doing? What is happening? That simply means that I am some kind of like using that our enzyme again called dehydrogenase. Who am I dehydrogenating? Malate. So this should be malate dehydrogenase. Genase. Cool. And in that process, I am using what? NAD plus to form what? NADH. But there's something, guys. Remember, we are supposed to use another enzyme here in this process called fumarase. Fumarase. So that is the enzyme I'm supposed to use here. Equally here, I'm supposed to use an enzyme called succinate dehydrogenase. So this enzyme is called succinate dehydrogenase. Okay, cool. Called succinate dehydrogenase. So from here we can see that the entire cycle is already completed because I'm dehydrogenating this guy to come back to this very compound. See, this is my carbon atom here. Here is it. This carbon atom here. Here is it. But we have removed what? This hydrogen. We have removed this hydrogen to be left with this carbon atom here, with this our carboxyl group here. So that is what is actually taking place here. Okay, guys, we can see that we have completed the entire Krebs cycle. Now from here, we want to look at what have we produced at the end of the day. You notice that as far as NADH is concerned, we have how many? We have one produced here, we have the second one produced here, and then all through we have the third one produced here. So how many NADH for one complete carboxylic acid cycle? There are 3 of them. But now remember that from glycolysis, where we had splitting, where we had dihydroxyacetone, from our glycolysis, remember we had the splitting of fructose 1,6-bisphosphate. Do we remember this compound? It splitted into 2. That was glyceraldehyde, 3-phosphate, and dihydroxyacetone phosphate. This one is running its own glycolytic pathway, while this one is running its own glycolytic pathway. So we are going to have one pyruvate formed from here and one pyruvate formed from here. So from one glucose molecule, we have what? 2 Krebs cycle. Are we there? Because this is from one glucose molecule that we are going to have this compound that is going to split into 2. So remember this. So everything formed here for a single glucose molecule is times 2. So if to say we have seen just for this entire Krebs cycle, we have seen just 3 NADH formed. So for a particular glucose molecule, one glucose molecule, during the process of Krebs cycle, how many glucose molecules are supposed to be there? During one Krebs cycle, how many NADH are supposed to be there? 6, right? 6, because we have 3 for 1. Now, going to FADH, all through this cycle, we have seen FADH just at which point? Here, where we are converting from succinate to fumarate. That is our hydrogen carrier in that case. So if you say that is actually what we have, so that means we have just one FADH form in one Krebs cycle. So for a single glucose molecule, we should have 2. So everything here is times 2. I'm saying so because when we are going to be entering the electron transport chain and then doing some calculations of the NADH being formed or the number of ATPs being formed, we will need to use this. So we have 6 NADH here. But if talking of this other part here, remember that we formed another NADH. This is another NADH formed here when we are transiting from pyruvate to acetyl-CoA, isn't it? So if we want to talk of that, so all together here we should have 4. And when we are multiplying by 2, instead of having 6, it should be what? 8. Are we there? So take note of this. So let's say for this entire cycle, we have 8 NADH formed because we have this one, we have the other one this way, when we are carrying out the hydrogenation, this one, we also have this one equally, and we have the last one at which point? Here. So there are 4 of them. So multiplying by 2. Since for a single glucose molecule we are going to have 2 Krebs cycles going on, we must have what? 8 NADH. Okay guys, I think that was really cool. I think that was really helpful. I mean, the video was really helpful, and I guess from this Krebs cycle we have really understood a lot. So stick with us, stay with us, and please follow us, hit the like button, and make sure you keep subscribing, and please assist us in whatever way you can.
1, 2, 3, action.
Hello guys, you are here with Salem, and today